Okie dokie, we're gonna go ahead and get started. I'm sorry if the audio is a little off. We're trying to figure that out here on the iTap end of things. Quick one announcement before we get started. I'll do a second announcement kind of halfway through. We have 2 mugs up for raffle. We have raffle tickets up here. Please grab one. They're MU Biology mugs with a shout out to George P. Smith, our pride and joy here at Mizzou. So we're starting off with Sean here, and typically Victor and I would try to think of puns, but I'm gonna go with a dad joke before he gets going.
Yes.
I went back home, and one of my younger cousins asked me if trees poop, and I said, "Where do you think number 2 pencils come from?" So there you go. I got Victor stamp of approval there. All right, without further ado, Sean Thomas.
Thanks for that nice introduction, Marty. Today I'm going to talk about how duplications, specifically gene duplications, drive diversity across plant life. Before we get started, maybe this will bore some folks. We'll have a little bit of a philosophy lesson. But some philosopher back a long time ago made the quote, necessity is the mother of invention. But I want to challenge that philosophy. And so, like, across human history, we've developed all sorts of crazy things like cars, lamps, airplanes, computers, our phones. And I can't really live without my phone. But people back in the day didn't have phones or cars or all these sorts of things. And so it wasn't necessary for them to live. But in this case, right, these innovations, I can't live without my phone today. These innovations are driving necessity. And so flipping that quote on over, invention, I think, is the mother of necessity. And so nowadays, I can't survive without my phone. I don't think anyone else in the audience who would be lost if they didn't have their phone. Raise your hands. Yeah, good number of people. And so we flip this quote on its head. And so, what is it that actually gives rise to invention? And so, if we think about how things are invented, someone needs to think about it. They need to have a lot of time on their hands in order to be able to create and invent things. And I think also a big part of that is coffee. In order to do the work that I do, I need a lot of coffee. And so things like time, coffee, having the freedom to be able to think and be creative is what I think is the mother of invention. And so I'll just read this quote real quickly. Man's creative genius flourished only when his mind was freed from the worry of daily toils and was permitted to entertain apparently useless thoughts. And so this may be a bit of a stretch, but I think redundancy is the mother of invention. And so when tasks like farming and making sure everyone is safe became redundant, right? We have farmers that do the farming, develop our food. When those tasks become redundant, we now have time and energy because of those redundant tasks in order to invent things. And so my philosophy is that redundancy instead of necessity is the mother of innovation. And so we'll get back to how that relates to what I'm talking about, but we also need to do a quick biology lesson for those who don't know. But all living things are made up of cells, and within those cells there are nuclei. Within the nuclei there are chromosomes, and chromosomes are made up of genes and DNA. And if you remember from high school biology, DNA is what gives the instructions for life. And so these are coded into genes, which give us our traits like hair color, eye color, and the traits that are in any living organism. And so, expanding upon that, the central dogma of biology is that DNA, which contains the instructions for life, gets transcribed into RNA, which is a messenger, which then gets translated into proteins, which do all of the functions that are necessary. So, metabolizing things, or being able to move around, being able to do all of the functions that we do are a result of proteins, which stem from DNA at the beginning. And so in my research, I'm interested in what happens when we have redundancy in DNA or duplications in DNA. And so people have explored this for decades at this point. And so the quote that I showed originally came from this book called Evolution by Gene Duplications, which was written in the 1970s. And this book posits that gene duplications or redundancy of genes plays a major role in evolution. And so duplications, summing this book up into one sentence, duplications have a major role in novel trait development. So from how crops are developed to human diseases and cancer and all sorts of things. And so thinking about duplications, thinking about this at the level of genes, we can have, for example, this original gene here that has these 4 different functions denoted by these colors here. When the gene is duplicated, we now have 2 copies of this gene that do the same thing or have this redundant function. But what can happen over time is that these genes will diverge. And so the sequence will change with random mutations, or parts of the gene will get lost, and we can have a couple of things that happen. And so for the most part, these genes are degenerated or lost. And so thinking back to our example of creativity, a lot of my creative thoughts are kind of garbage, right? They can just get thrown away. And across everyone, right? Everyone has had just random creative thoughts that, for the most part, mean nothing. And so the vast majority of genes get lost because they're not useful. But sometimes what can happen in the case of neofunctionalization is that these mutations will give rise to new functions in that redundant gene. But also what we can see is this pattern of subfunctionalization where the genes now have partitioned the different functions across the 2 duplicates. And so this is generally, very simply putting, kind of the fate of duplications as we see from evidence in biology. And so duplications can arise from a variety of different mechanisms. They can arise from what we call small-scale duplications, and these are duplications of single genes or small segments of DNA. And for those who are interested in how these actually happen, here are some mechanisms. But we also have what's called polyploidy, or whole genome duplication. And this is where the entire DNA content, or all of the chromosomes, become doubled or tripled or multiplied in many times. And so in the example of a whole genome duplication, we have, right, this diploid organism. So jumping back to high school biology, most organisms are diploid, meaning that they have 2 sets of each chromosome, one from their mother and one from their father. And what happens during the case of a whole genome duplication event is that now we have double that total amount of DNA, or a polyploid or a tetraploid in this example here. And so when we look at polyploidy, polyploidy is prevalent across plant evolution. So all of these red dots that you see here in this green part of the tree, these are all whole genome duplications that have happened across the evolution of plants. When we compare this to animals and fungi, there are not that many. And so plants have this interesting ability to be able to retain these duplications and be able to use them in their evolution. And so, jumping to a few examples, so who had coffee this morning? Raise your hand. Yeah, pretty much everyone in the room. Coffee is an example of a polyploid. And so these 2 species, Coffea canephora and Coffea eugenoides, the 2 genomes from these 2 species make up Coffea arabica, which is the coffee that we drink. So if you enjoy lattes from Shortwave, shout out to Shortwave, they're the best coffee shop in town. Then you're drinking Coffea arabica, which is a polyploid. And fun fact, I don't know if there's that much evidence for this, but we know that this species of coffee has high caffeine content, and then this species of coffee tends to have really good flavor profiles. And so the hypothesis is that the caffeine content in coffee that we drink comes from this species, whereas the flavor that we enjoy, nutty, chocolatey flavors, if you like Ethiopian coffees, floral and fruity flavors, comes from this species. And so the combination of the two makes the sum better than the parts. So going to lunch or snack time, maybe you had some strawberries and peanuts sometime today. If you had strawberries are an octoploid. So they have 8 sets of genomes within their singular genome. And peanuts are another example of a tetraploid like coffee. And then today, we're all at ITAP. I think people are enjoying beers today, so cheers to that. But what is beer made of? Beer is predominantly made of wheat, which is a plant, if you didn't know. And it's also made with yeast, which does the fermentation. Yeast is not a plant, by the way, it's a fungi. It's one of the few fungi that have had polyploidy in its evolutionary history. And so the combination of 2 polyploids gives us the beverage that we're enjoying tonight. So I think that's quite cool. But going to what I research, I study polyploidy in brassicas. So if you didn't have strawberries or peanuts today, maybe you had some kind of brassica for lunch, or maybe you'll have it for dinner. But things like broccoli, cabbage, cauliflower, kale, kohlrabi, canola oil, pak choi, napa cabbage, turnips. These are all examples of brassicas which have experienced a lot of polyploidy in their evolutionary history. And so all brassicas share this hexaploidy event, or share a hexaploidy, which means at one point in their evolutionary history, their genomes tripled. And then in the case of like canola oil and rutabaga, they had additional polyploidy events, or this tetraploidy that we're seeing here. But from other data, we know that brassicas, right, this specific species, these crops, aren't the only things that have this polyploidy event. And so my research focuses on what other species share this other polyploidy event. And so I have some data. This might be a bit of a confusing figure, but this hexaploidy is not unique just to the brassicas. And so we have brassica here, and we have a bunch of other species that are represented represented in green here. And so everything that's green here also shares this hexaploidy event. And we have evidence for this using these plots here. And so what we can do is we can understand, across the x-axis we have what's called sequence divergence. And so as sequences diverge over time, right, we talked about the example of when we have a duplicate, they diverge over evolutionary time and can gain mutations. And so across the x-axis we're tracking the mutation rate of different genes, and then on the y-axis we're looking at the counts of those. And so when we have a polyploidy event, what we expect to see is a peak that forms. And so the peak that's associated with the hexaploidy event is this peak here. The peak that we see at the lower end of the chaos is associated with what's called small-scale duplications, which we talked about earlier. And so if we look across all of these green species, right, we see that they share this hexaploidy peak that's common with the brassicas. And so from this evidence and other evidence, I won't get into the details of it, we have figured out the boundaries of when this hexaploidy had occurred. And then, right, these species are ecophragmous, and the outgroup don't share this pattern, and so we're able to tell that they don't have this polyploidy event. That's all cool, right? Nerds like me that are interested in how genomes have evolved think that's really cool. But what do these duplications, how do they influence traits in brassicas and other relatives? And so one big thing that's been studied a lot is domestication. And so we like to think of brassicas as the dogs of the plant world, because just like dogs, which come in all sorts of shapes and sizes, brassicas come in all sorts of shapes and sizes as well. And so genes from polyploidy play a big role in domestication. And if you want to look at all the data, you can read this paper here, which was done by my lab. But they also influence things like flavor. So glucosinolates are a secondary metabolite which influences the flavor of mustard. So things like mustard, wasabi, horseradish. If you've had arugula, right, it has a bit of a peppery flavor. That peppery flavor comes from glucosinolates. And originally this was used as a defense mechanism against insects. But because humans like that flavor, we've now developed them into all these different varieties of things. And if you want to read more about how glucosinolates are involved in lubrications, you can read this paper here. But I'm interested in not the crop species, but kind of the weird wild relatives that occur. And so one of those species is called Cacalia myrtema. And this is this plant that grows on these coastal dunes. It looks like this in the wild. And these are the really bad, kind of dying plants that I grew in the greenhouse. They look okay sometimes. They're very pretty, I think. But these plants have originated in the Mediterranean, and so this is where they originate. But because they're salt tolerant and because their seeds are, they don't sink in water, they're able to float. So if you see, this is the seed of Cacalia. It kind of looks like a speedboat. It's able to float over on ocean water, and it's colonized the coasts of the U.S. and Australia and parts of South America. And so that in and of itself is interesting, but that's not what I'm going to talk about. But because it grows on coastal dunes, it's also salt tolerant. And so I have some data here. I'll kind of go through this figure, but duplications are influencing salt tolerance in Cacalia. And so all these terms here are different functions associated with salt tolerance. And what I've done here is look at which kinds of duplicates are associated with these kinds of functions. And so things like amino acid metabolism, cofactor metabolism, lipid metabolism, lots of different metabolisms, transporting sodium and other terms are involved in how plants can be tolerant to salt. And so when we look at single copy genes, which are genes that are not duplicated, there's only a few functions that are enriched in terms of salt tolerance. But the vast majority of functions have been duplicated, and these duplicated genes play a role in how Cacalia is salt tolerant. And so that's all the data that I'll show for today. But why is salt tolerance important? I just kind of want to leave this with you. So across the world, right, there's a lot of different salty and saline soils, and high soil salinity makes lands infertile. And so things like harsh agricultural practices, deforestation, and climate change lead to increased salt in soils, which is a bad thing. Because, right, as of now, 24% of all irrigated lands are affected by salt. So, and this leads to economic losses of over $27 billion annually. And so, right, if we can either create salt-tolerant plants or make current crops more salt-tolerant, we can, right, adapt to this process. So we either need to stop the processes of climate change, which, given our track record, is probably not going to happen, or we need to find ways to adapt to climate change. And so, right, through resilient crops and or developing new crops using species like Cacalia, maybe we can develop more crops that are resilient to salt and other abiotic stresses like drought, heat, and other things. And so I'll end my talk there, but hopefully some of the things that you may have gotten from this talk are that duplications are an important evolutionary force. Plant polyploids are everywhere. We're drinking some of them today. And duplications in plant diversity can help us to develop stress-resilient crops in order to adapt to climate change, which we cause. And with that, there's a long list of people I need to thank, and I'll take questions.